what purines are, why they are important. I'm also going to tell you guys about some of the differences between purines and pyrimidines, and more specifically the differences between purine and pyrimidine synthesis. And I'm also going to tell you guys about some of the drugs that are also used to inhibit purine synthesis as well. So to get started, what are purines? Well, purines are, as pyrimidines are, they are nitrogenous bases. They are 6-membered rings, but they are fused to a 5-membered ring as well. And purines are the adenine and guanine are purines. Now, here are the structures for adenine and guanine. Now, as you can see, the main difference between purines and pyrimidines are that purines are 6-membered rings fused to a 5-membered ring, whereas pyrimidines are just 6-membered rings. As I mentioned before in the pyrimidine synthesis video, that pyrimidines are easy to remember by the mnemonic cut the pyramids. So CUT, cut the pyramids. So think about that, guys, when you if you're ever confused about what is a purine, what is a pyrimidine. Now, a kind of a way I remember which is 6-membered fused to a 5-member ring is that I always think purine, pure. I always think, for some reason, it helps me remember that purine reminds me of pure, which reminds me of something that's more complete or more complex. 6-membered fused to a 5-member ring. So what are purines used for? Well, purines are used for DNA synthesis. They are also used as energy carrier molecules such as ATP and GTP, as we all know. It's also used for something very important known as S-adenosyl methionine, or SAM, which is used in some of the biosynthetic pathways for certain hormones. Now, purine synthesis requires several things, just like pyrimidine synthesis does. It requires glutamine. It requires glycine. Now, this is a big difference between purine and pyrimidine synthesis, is that purines require glycine for their synthesis. Purines also require aspartate and tetrahydrofolate, vitamin B9, just like pyrimidine. So again, vitamin B9 is very important for both purine and pyrimidine synthesis. So how does it all start? Again, well, we start with the pentose phosphate pathway in purine synthesis, and pentose phosphate pathway produces ribose 5-phosphate, which then gets processed into 5-phosphoribosyl 1-pyrophosphate, or PRPP, via the enzyme PRPP synthase again. And then, as I mentioned before, PRPP synthase is inhibited by ADP and GDP and activated by inorganic phosphate. Now, this is where it gets a little bit different, guys. As we learned in pyrimidine synthesis, PRPP actually gets utilized directly, but with purine synthesis, it needs to be processed further. Now, the purine synthesis pathway, one of the main enzymes, one of the most important enzymes in the pathway is this enzyme, glutamine PRPP aminotransferase. And what it does is it actually utilizes glutamine to add an amine group to the PRPP to result in 5-phosphoribosyl 1-amine. In the process, it converts glutamine to glutamate, and as I mentioned before, this enzyme, glutamine PRPP aminotransferase, is the first committed step. It's the most important enzyme I want you guys to remember for purine synthesis, because it is the first committed step in purine synthesis. Now, this enzyme is actually inhibited by AMP, GMP, and IMP, some of the products of the purine synthesis pathway. And the next several steps, I'm just going to quickly go through what is required for the step. I'm not going to actually get you guys to remember all the steps because it's very complex. There's actually 14 steps in the purine synthesis pathway, but all I want you guys to know is what is required for the pathway. So the first step after the 5-phosphoribosyl 1-amine, you require glycine and ATP. Now, this is where glycine is important. The next step requires tetrahydrofolate. The next step requires glutamine. Another step requires ATP. Another step requires CO2. Another step requires aspartate and ATP again. Another step requires tetrahydrofolate or derivative again, and all leading to a dehydration reaction and the production of inosine 5' monophosphate, or IMP. Now, you guys may want to look this up on your own to see each step, but really the important point I want to get across is that what is needed or what is required for the purine synthesis pathway. And as I mentioned before, glycine is required, glutamine, aspartate, and N10 formal tetrahydrofolate, and a couple ATPs are all required for purine, the purine pathway into the production of IMP, or inosine 5' monophosphate. Now, as I mentioned before, the formal tetrahydrofolates, or tetrahydrofolate, is actually produced by dihydrofolate reductase enzyme, which is actually inhibited by methotrexate and imidopterin. And this is where these drugs will actually act in the purine synthesis pathway to suppress the purine synthesis, and it is used, as I mentioned before, methotrexate is used as a common chemotherapy drug and as utilized for medical abortions as well. Now, once you have IMP, what can happen is it can be IMP can actually be processed into AMP, utilizing the amino acid aspartate. So an easy way to know that, easy way to remember that, is IMP to an AMP. Aspartate starts with an A, and AMP starts with an A. That's how I remember it, guys. So IMP to AMP requires aspartate amino acid. And then AMP can actually be processed to ADP, which AMP can process to ATP. IMP can then also go down a different route, a different pathway. It can actually be processed into GMP via the utilization of the amino acid glutamine. And again, that's an easy way to remember it, guys. GMP, glutamine. Glutamine starts with a G, so that's how I remember. That's just an easy way to remember which amino acid is required. So once you have GMP, what can happen is the GMP will be processed into GDP by the enzyme guanylate kinase, and then GDP will be processed to GTP. Now, AMP and GMP can also come from a different source, and that source is the purine salvage pathway, and I'll talk to you guys about that in just a bit. But just remember, guys, that AMP and GMP can also come from a salvaging pathway as well. So once you have ATP and GTP, they can be processed in a couple of steps and be utilized for DNA synthesis. And another thing I want to mention is that inosine 5-monophosphate can actually be metabolized into something known as hypoxanthine. And hypoxanthine, via a couple of steps utilizing the enzyme xanthine oxidase, can be actually metabolized into uric acid. And that is very important in human health, that we'll talk about in another video. So I mentioned the salvage pathway. Now, what is the salvage pathway? What is the purine salvage pathway? Well, when you have hypoxanthine, which is derived from IMP, or guanine, what can happen is, and guanine, by the way, guanine is actually a metabolized product of GMP. Now, what can happen is hypoxanthine can actually be reprocessed into IMP utilizing PRPP, and guanine can be reprocessed into GMP, again with PRPP. Now, a common enzyme does both of these reactions, and that enzyme is hypoxanthine guanine phosphoribosyl transferase, or HGPRT. Now, that's the enzyme I want you guys to remember, that hypoxanthine guanine phosphoribosyl transferase, or HGPRT, is the important enzyme in this reaction, and that is actually an important enzyme in certain genetic disorders as well, and I'll mention that in another video. So once you have IMP and GMP, IMP can actually be converted into GMP or AMP. AMP and GMP can actually be, again, brought back into that pathway I just showed you guys in the previous slide, and then actually processed into ATP, GTP, and then into utilized into DNA synthesis, or into utilization as energy-carrying molecules. Now, the important things about the purine salvage pathway that I want you guys to know is that the purine salvage pathway requires PRPP, which is, again, a product of pentose phosphate pathway. It also decreases purine degradation. Now, this is very important, and I'll tell you guys why in the next lesson, is that because you are salvaging these products, you're salvaging hypoxanthine, you're salvaging guanine, they're not being metabolized. They're not going to have to be metabolized and degraded into some of the other toxic products, such as ammonia or ammonium, and the other toxic product, which is uric acid, which I showed you in the previous slide. So because the salvage pathway actually decreases purine degradation, it's very important in human health. And it's because it decreases uric acid. That's one of the main things it does. So it actually decreases uricemia, or blood concentration of uric acid. So in patients with hyperuricemia, if they have high levels of uric acid in their blood, this pathway may be affected in some way. And now I'll mention this again in another lesson as well. So all I want you guys to remember is that this pathway is critical for maintaining proper uric acid levels in the blood. And in some cases of hyperuricemia, this pathway can actually be affected, and in some genetic disorders, this pathway can be affected. And I'll show you this. This will be further talked about in the purine and pyrimidine breakdown video as well. So, anyways, guys.